to the Mind and Matter podcast. I'm your host, Nick Jikomis, and today I'm speaking with Dr. Amy Reichelt. Amy is a neuroscientist by training. She received her PhD in neuroscience from Cardiff University, and her expertise is in the behavioral and cognitive neurosciences, in psychology, and aspects of molecular biology. She has led research that sits at the intersection of neuroscience and nutrition, and she's really interested in uncovering the neural mechanisms that underpin how diet actually impacts brain health and cognition across the lifespan. So Amy and I talked about topics in that general area, how our brains are built to learn and remember things, and how that influences our ability to learn and remember where food is and how to get it and all of those associations. We also talked about how diet itself, the composition of our diet, can impact circuits in the brain, not only acutely, but how it can cause them to change in various ways. So we talked about experiments done in rodents, giving them a so-called Western diet, literally giving rodents food from the grocery store that's high in saturated fat and sugar that a lot of us eat, and seeing what effect does that have on their general health, but also what is it doing inside of their brain and their ability to actually perform learning and memory tasks. So there's a lot of fascinating stuff there about how the diet actually causes physical changes to happen in the brain. We touch on topics related to neuroinflammation. We discussed everything from omega-3 fatty acids and different foods that can have either a positive or negative effect on brain health. We talked about psychedelics and their impact on neuroinflammation and how that might be relevant for things like diet-induced obesity. We talked about the parallels between food addiction and drug and other forms of addiction. And we talked about a lot of the mechanics of what actually goes on inside the brain at the cellular level with respect to some of these phenomena. So if you're interested in how diet and nutrition impacts brain health, this will be a really interesting episode to tune in for. As always, if you like the content on this podcast, please do like, share, and subscribe either on social media or just by telling a friend about the podcast or an interesting episode that you listen to. You can also subscribe to my free weekly newsletter at mindandmatter.substack.com, where I provide updates about the podcast, including upcoming guests, alert you to other content I've written either on my Substack or elsewhere, and provide updates on what's going on in the research world in topics related to what I discuss on the podcast. You can also like and subscribe to the video version on YouTube if you just Google my name or Mind and Matter podcast. And you can support the podcast by visiting any of the links that I provide in the episode description. You'll see those down below. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. And with that, here's my conversation with Amy Reichelt. Dr. Amy Reichelt, how are you?
I'm very well, thank you.
Can you start off by just telling everyone a little bit of who you are and what your scientific background is?
So I have a PhD in neuroscience. which I obtained from Cardiff University in the UK. Before that, I did a bachelor's in psychology from University of Birmingham. I really first cut my teeth into what went on to become one of my major research focuses over the past decade during my honours as an undergrad. And I got to examine how the endocannabinoid system and the cannabinoid system affected palatability of solutions of sugar in rats. So utilizing some of the then quite novel cannabinoid antagonists, the CB1 rimonabant and associated molecules. And I then kind of put that aside and went on and did my PhD, where I really looked at behavioral neuroscience and how the tauopathies and Alzheimer's models of dementia affected frontotemporal function using various mice. After that, I went to University of Birmingham for a postdoc and studied memory reconsolidation, kind of riding out that really fun wave and really looking at how appetitive memories, so memories of food, could be updated using rats. And that was an awesome adventure working with now Associate Professor Jonathan Lee. But I kind of wanted to escape from the UK, and I'd always been a bit of an adventurer, and decided that I was going to go to UNSW in Sydney, where I worked in both the School of Medical Sciences and the School of Psychology, working with Margaret Morris and Professor Fred Westbrook, and looking at how food addiction and memories for food and memories in general were affected by food. And this really got me so interested in how our diet and obesity could potentially be impacting not only our bodies, but our brains, and effectively controlling aspects of our behavior. And I was really fortunate while I was at UNSW to secure Australian Research Council funding in the form of a DECRA fellowship. And that allowed me to start my own research program and really start to hone down into some of the ways that our diet can control effectively our behavior to food and just in our environment in general. So I became really interested in how diet can influence memory formation and also behavioral control. And when I look back at that, it was, an amazing time. I was living in Australia, and I then went to RMIT University as a lecturer, so an assistant professor equivalent, and continued my research there. And I then kind of went, I've kind of done Australia for a bit. I'm going to go to Canada. And went and worked with Tim Bussey and Lisa Saxida in the lab at Western University in London, Ontario. And I was working there, utilizing a lot more sort of cool genetic tools like chemogenetics, optogenetics, and fiber photometry to be able to hone in a bit more on what these diets are actually doing at the neuronal level, beyond just the molecular biology and the behavioral changes, like actually being able to control things. I was really interested at the time in how the extracellular matrix was controlled in potentially neuronal communication, and did a lot of work there on how our diets can then impact just specific sets of cells. And then COVID hit, which was a thing and still ongoing. I secured a position back in Australia at University of Adelaide. However, my partner was still in Canada, and ended up trying to do science remotely. Wasn't ideal, and then decided, hey, maybe I should head back, and came back to Canada. And I'm now working as director of neuropharmacology at Cybin, who are a psychedelics pharmaceuticals company. And it's been a massive learning curve over the last couple of months, but it's still exciting. I also started my own business called Cognition Nutrition, and that's really being able to utilize my understanding of brain and nervous system and also behavioral control, and combining that with my love and interest in nutrition.
Interesting. So your background is really mainly as a basic research scientist, dissecting various aspects of feeding behavior and how that impacts the brain. And there are a number of things I want to talk about here, but I think one of the things that someone like you will appreciate is how, the brain and the entire body is really one integrated apparatus that does all sorts of interesting things. But with respect to feeding, I'm wondering if you could walk us through some of the basics of how a normally functioning brain, how a brain that's healthy helps orient an animal in its environment so it can figure out what to eat and when to eat. So how, I know that there's a lot of things that we could discuss there, but if you could give us kind of a neuro and physiology 101 there, what is the brain basically doing at a very high level to get an animal to find food in an appropriate and adaptive fashion?
Well, what I find really fascinating is that although as humans, we think that we're so much higher order animals compared to the rats and mice that I'd be working on in the lab, we effectively have a lot of the same neurotransmitters and the same systems within our brain. And what really it comes down to, when we think about how our little rat in the wild versus us as, potentially in the past is, our caveman ancestors, we were all, needed food for survival. And again, this falls back to a lot of thinking about, associative learning. So that sort of psychology second year course that everyone found a bit difficult because we start thinking about things in a bit more of an abstract way, but really what happens to us in the real world is we find something that we find rewarding, and food for us is definitely a reward, particularly our palatable foods like cakes, chocolates. They're high energy, and we need these for survival. And our brain is still hardwired the same survival way. So, we have our brain's dopaminergic reward signaling system, and this reward system that generates basically a prediction error when we're in, when we find something that's surprising and interesting, be it, suddenly eating something that's delicious, and finding one of these little hole in the wall cafes when you're traveling somewhere. We suddenly learn a lot about our environment, and we encode all of this information at once. It's like, this is the location, so that's requiring our hippocampus to have learned that location. Our reward system is firing, so that's requiring our dopaminergic system, and we have our emotions associated with it. This is really good. So, we have our prefrontal cortex that's kicking in, behavioral control, being like, should I buy another one of these amazing cakes? And this is like our simple system, but in the same way, we can model this in the laboratory when we, have our rat that might be, in a tea maze, and it learns the direction that it has to go to find food by geometrically encoding the landmarks around it that direct it to go to the right place. And I think about this in the same way that, when you're in the supermarket, you learn where the sweets aisle is very quickly, and you learn how these certain rewards make you feel good. And nowadays, however, we're in an environment that is completely replete with all of these kind of rewarding foods and beverages, and it's really difficult for us to override these innate predispositions to want these certain foods, because we know that they make us feel good. We know that they're rewarding.
So one of the terms that you used here was palatability. So basically how good something tastes. And what's really interesting is that in many aspects of palatability seem to be hard coded, right? Many animals will generally find certain things to automatically be very palatable. And it happens to be, for most people, most of the time, that the most palatable foods are oftentimes what we would consider to be the most unhealthy foods. They're very calorically dense. They're filled with things like sugar. Can you connect the dots there a little bit more for us? From an evolutionary perspective, why is it that foods like that are more or less automatically so highly palatable to people?
Um, I mean, these sort of foods, they trigger not only our dopaminergic reward system, but they also have impacts on our other endogenous neurotransmitter systems, like our endocannabinoid system, which again is involved in palatability, our opioid system, which again is linked to the sort of hedonic aspect of a reward. And these foods, because they're high energy, are important for survival. And our brain being hardwired for survival makes us like these things that help the species continue, in the same way that our reward system is triggered by sex and sociability and interacting with other people, that we crave these kind of behaviors and this reinforcement through our brains neuronal circuits are what is definitely driving this sort of ways of prolonging humanity as a species, in the same way that our little rat in the wild is just looking for a mate and looking for a good source of food. And I think that it's we simplify our behavior now back down. We think that we're in these situations where everything is so much more higher than really we are sometimes, because when it comes down to it, we're still looking for rewards. And we know as well that the reward system becomes hijacked by drugs and abuse. This is where we're really turning up the reward system to 11 and making everything so much it's really being hijacked by reward. And this is making people crave certain substances, in the same way that you learn about foods that are typically the really highly palatable foods, fat and sugar, and that these foods the things that we know when we're stressed can make us feel better because we get that dopamine kick. We also know that when we're stressed, our brain requires more glucose. And we also have learned that these foods physiologically are a quick way of getting glucose into our bloodstream and then into our brains. So that's why, when we're stressed out, it can make us really crave these kind of high energy, high, particularly sugar foods. We want these quick kicks of sweets and chocolates, and our stressful lives can sometimes encourage this. And also we've got these behaviors that kick in. So we learn about how things make us feel, but we can also start to when we continue these behaviors, we start to form habits, and habits are really unconscious. And it can be eating when you're not hungry, or always having dessert because you always have dessert, or eating lunch because it's 12 o'clock, even though you maybe got up late and had breakfast at 10:30. Just these kind of behaviors where you start really overriding your own physiology. And I was talking to a client the other day, and I was like, it's so almost uncomfortable for us to feel hungry nowadays and be able to really feel that, that sense of I'm really starving hungry and just want to eat. Because we're in this environment where we habitually graze upon things. Working from home is the worst. I think I just hang out in front of the fridge for at least 15 minutes a day, just sort of staring into what I know I've bought and put in there already. It's not going to manifest me some food magically. But these just sort of routines that we get into. And also my research has really suggested that our diets do affect these key areas of the brain that are involved in cognition, such as our prefrontal cortex and our hippocampus, as well as potentially also our brain areas that are involved in emotional learning, such as the amygdala. Mm-hmm. And this has been sort of a big line of work between both my lab when it was based in Australia, but also other labs in Canada and the US that are all really interested in looking at how diet can fundamentally change the functioning of these brain regions that are so important for us in behavioral control and learning.
One of the things that I think is interesting to get into here is, on the one hand, it makes perfect intuitive sense that, as animals that have evolved in the real world, our brains are naturally tuned into, using general purpose learning and reward mechanisms to figure out how to navigate the environment, among other things, to find food and survive. So it's not going to be a surprise to anyone that, well, of course, our brains learn to correlate and associate the taste of something with what it looks like, or where it's located, or what we had to do to get it. And we sort of figure out all of those things. But, based on what you were saying and based on, the kind of story we always hear told, if we're just going to use sort of the cartoon evolutionary psych explanation for why we have many metabolic problems today, it's that our brains evolved in a state where abundance was the exception rather than the rule. So it wasn't every day that a caveman was going to find some highly calorically dense sweet foods the way that we can go get them whenever we want in the supermarket today. And so today, in a state of constant overabundance, there are not a lot of good stopping mechanisms to prevent us from just overgrazing and overindulging. And that brings us to the flip side of this equation, which is, the brain is capable of learning and making associations with respect to food and other things. But you started to basically say that what we eat and the patterns with which we eat can also literally affect circuits in the brain. And so I'm wondering if you could start to talk about that side of the equation. If we think about modern diets today that are highly calorically dense and high in sugar and other things, and I guess I'll let you define what the modern Western unhealthy diet really is. How does the ingestion of that kind of diet actually then start to go into the brain and literally affect circuits there?
Yeah, and so sort of to define our modern day diet, a lot of research tends to call it the Western diet. When we think about it, it's full of ultra processed foods, our convenience foods. It's high in saturated fats, and it's high in our refined sugars such as sucrose, but then also more sort of nefariously are even faker sugars like our high fructose corn syrup and these kind of hyper sweet foods. And these foods, we know are completely ubiquitous within our own environment. They're so easy to obtain, and they're not only having effects on our bodies when we overconsume them. So for instance, over Christmas, we all tend to overindulge a little bit too much, and potentially Northern Hemisphere where the weather is tends to be bad. We're not getting outside. We're not doing much. So we tend to overindulge across a certain period of time. And by January 1st, we all start to look at ourselves and we're like, I feel kind of gross. My brain isn't really ready to go back to work. I haven't been to the gym for ages. I'm feeling very sort of sluggish. And potentially, it's these sort of periods of time where we do overindulge and do overconsume these foods, even in spite of typically having, a generally quite healthy diet for us, that we start to notice that, these foods might be starting to affect our mood and, how clear our mind is. So what I think is really interesting is that in sort of 2013, 2015, between 2013 2015, Margaret Morris's lab at UNSW conducted some experiments that were giving rats access to our own version of high fat, high sugar diets, our Western foods, which were literally Western foods. So we used to go to the supermarket and buy these rats like pies and cakes and cookies, and big white spraggly rats would eat these foods and they would gain weight. But what was actually found is that within just a period of 4 to 5 days, these animals started showing deficits in hippocampal dependent learning tasks, particularly spatial learning tasks as spatial recognition, which is really dependent upon the hippocampus. And this was before the onset of any kind of major weight gain in these animals. And it was really fascinating because it started to strike me that there's something going on in the brain that's even faster than just our original concept, which was you gain weight, you gain adipose tissue. Adipose tissues leads to the release of cytokines into the bloodstream. That's then going through into potentially through the blood-brain barrier into the brain, and then that can start to cause changes. But there's very rapid effects of these diets that is potentially before even the onset of adipose tissue gain. And
so just to make sure I'm following, you're saying you would have rats on some normal rat diet, and in a moment we'll talk about what that was. You literally start giving them food, human food from the grocery store. And when you say high fat, high sugar, this means in particular high saturated fat, and high sugar just means sucrose, high fructose corn syrup, like all the stuff that you find in human supermarket food. They love to eat that stuff, and they will choose to eat it just like we do, and they will gain weight just like we do and become heavier. But what you're saying is when you give them tests to measure their like spatial abilities and other cognitive abilities, they actually start to perform worse on those tests within just a few days, which is well before they actually get heavier.
Yeah, so it's kind of mind-blowing when we think about how our diets can so quickly affect our brain function. And also this kind of hit home to me when it came to that sort of thoughts about, how sometimes you might just have a really big weekend and you might eat quite healthily in the week, but then potentially, there's stuff going on, at the weekend, and then, 3 or 4 days of eating unhealthily, maybe having this ongoing effect. So looking at, that, these diets obviously have very striking effects on cognitive abilities when it comes to spatial memory. And so that's very sensitive to the hippocampal function. And I became really interested as well in how our high sugar aspect of the diet, because rather than just looking at something as being a high fat and high sugar diet together, but teasing apart that potentially there is an impact of sugar. And we were giving rats access to 10% sucrose solution, which is the same as about a can of soda in terms of the percentage. And we were binging the rats as well on the sucrose rather than just giving them open access to it. So what was really interesting in these studies was that the rats would titrate their calories almost. So they would restrict down their chow consumption. So this would be our sort of normal rat lab chow, wheat bix, kind of boring pellets for them. So they would cut down eating that in lieu of, then filling up their calories with the sugar solution when it was allowed and put into their cage for just 2 hours a day. See these rats, they would just like run over. They've got water the rest of the time, but they were just like, yeah, it's sugar time. And I was really interested because, these animals weren't any different in terms of body weight. They just had access to sugar solution for 2 hours a day. And when we conducted some different sorts of behavioral tests on them, so we did the spatial navigation tests, and they showed deficits there. In one of the papers, we used a test of pattern separation, which really relies upon neurogenesis within the brain. So these animals were on for 28 days in these diets, and then we were using a more complicated spatial test with them where we could control the locations of the objects within an environment. So we could either make the objects far apart so they were more distinct, or we could bring them closer together to make them less distinct, and so put more strain on the hippocampus. And what we found in this case was that our rats that binged on sugar each day showed impairments when we made the task difficult, so made the separation much smaller. So when they came to then finding which was the novel located object, they weren't as good. But in basically the exact same setting, when we made the objects further apart, so putting less strain on the hippocampus, they could perform the task. So what made me think is that, we have people who are consuming these diets, which may be, very high sugar, and the effect of the diet, isn't as blunt as, just walking around being completely lost in the world, but they're very sort of insidious effects that may particularly, then be linked to a reduction in neurogenesis in the brain. We looked at proliferation markers, and we also looked at double cortin for our differentiating neurons, newer neurons. And what we found is that, only when we start to really tax the hippocampus do we see these differences. And I think this is really important, because when we think about, how we might function fine on a day-to-day basis, we have our normal routines, kind of do our own thing. But, when we start to really push our cognitive abilities, are they potentially being, detrimentally impacted by these diets?
Yeah. Well, what this immediately makes me think about is, well, there's a number of things to connect here, but, the concurrently with having this overabundance of highly dense, highly palatable food available at all times to ourselves, the progress that comes with civilization, that comes from technology and stuff, allows us to not push our cognition to those upper limits. And so more and more with time, even within my own lifespan, right, there's just less and less pressure. There's less and less need to sort of force yourself to use your brain to its full capacity, so to speak. And based on what you're saying, the deficits that you're talking about are almost masked by that fact. So let's back up a second. And I want to make sure I'm following what you were talking about before. So with the rats, what you were saying was you give some rats access to sucrose for just a couple hours a day or something like that. And what they would do is they would just start choosing to eat less of their normal food. And so they were consuming the same calories per day as rats that did not have access to sugar, but now a higher percentage of the calories that they're getting are coming from sugar. And then you're doing these other tests of some kind, and they're performing worse, but not if the tests are relatively easy. They're only performing worse if they get relatively difficult. That's the basic result.
Yeah. And what's really fascinating is that there's so many neuropsychiatric and neurocognitive assays that we have as people and that we can do as well, but we can back translate them for use with the rats as well. So another major study that I conducted was we used a version of the rat Stroop task, or the human Stroop task, where you've got both. So in general, the Stroop task, you have your stimulus, which has typically a word, be it red, and red is written either in the color red, which is your congruent stimulus, or you have an incongruent stimulus, which would be red written in blue. And when it comes to your Stroop task, you need your prefrontal cortex to be able to override your predisposition to just automatically read the word, but also follow your demand that is given to you. So it could be name the color of the ink. You have to say the color of what the word is written in. So when you have your incongruent stimulus, you get this conflict in your brain where it's going, I really want to say red, and you have to override that and say blue. So we had this task with rats that again reflects this kind of discrimination behavior. And again, we found that the rats that were binged on sugar performed worse in the task, but only when it came to the incongruent stimuli. And again, that's suggesting that these diets are having not only effects on the hippocampus, but the prefrontal cortex, which is so important for us to be making decisions. And then I went in and looked at the brain's post-mortem of these rats and did some various immunohistochemistry stains. And one of the key stains that I wanted to look at was parvalbumin interneurons. So these are our GABAergic interneurons that are really integral components of brain systems for like higher order cognition. They're balancing the inhibition and excitation in the brain. I think of them as being, those little mediators that you need to control your behavior and override certain responses because your neurons are firing. And also changes to these neurons in terms of their function or their populations are seen quite commonly in neurological conditions such as autism spectrum disorder or schizophrenia, but in a pathological way. But what we also found was that there were reductions of these neurons in the prefrontal cortex, but also the hippocampus. And this really fascinated me because this is showing that these diets are starting to have an impact on different neurons, key neuronal subtypes, and beyond just sort of neuroplasticity and potentially neurogenesis that's happening in the brain, that they could be having key changes to the brain and how it's functioning on mass. And I've been trying to figure out what's really going on, and I think inhibitory signaling is potentially one of the key ways that the brain is starting to become disrupted. And these diets as well don't just affect neurons, they affect the glial cells that are in the brain as well. So one of the other observations with exposure to high fat, high sugar diets, high fat diets, high sugar diets, is that you see an increase in microgliosis. So the microglial cells become activated as well. And that's suggesting that, there's inflammation in the brain. And some great work by Scott Konoski also showed that exposure to high fat and high sugar diets changes the permeability of the blood-brain barrier. So our blood-brain barrier is so important to preventing pathogens entering into our brain and starting to disrupt neuronal function. But if that becomes leaky or compromised, then all kinds of cytokines and chemokines and molecules are going to get into the brain and start wreaking havoc. And it appears that the hippocampus is quite closely related to the blood-brain barrier. So that's potentially why we see changes to the hippocampus so quickly. But also areas like the hypothalamus, we see changes in inflammation. And this has also been seen in pathology from humans, which I think is really exciting, particularly in the hypothalamus. And so this neuroinflammation is setting up. And then we've also got changes going on to neurogenesis and neuroplasticity, and markers of neuroplasticity are changing the interneurons, which I'm particularly interested in because our parvalbumin interneurons, high energy, they're fast spiking, they require a very sort of controlled environment. And what I was really interested in is potentially as well this interaction between, how microglia, when they become activated, could be detrimental and damaging pathologically specific neurons. And one of the other key areas I was looking at was that the parvalbumin neurons are surrounded by a specialized form, often of extracellular matrix called perineuronal nets. And if you just want to Google what a perineuronal net looks like, they're stunning. If you look at them under a fluorescent microscope, they're these beautiful little web mesh-like structures that surround neurons, and they perform multiple roles involved in plasticity, but also in protection of our neurons. So they form this microenvironment around particularly fast spiking into neurons like our parvalbumins, because these are very sensitive cells, and they not only stabilize connections through the synapses, but also can prevent oxidative stress damage from settling into these neurons. So I became really interested in whether or not these diets were also potentially being impacted by our perineuronal nets. And
yeah, so I think one of the themes that I'm hearing here is these perineuronal nets that you're describing are effectively these little mini protective shells that live around certain neurons and certain neural connections, and they sort of protect them from damage. They're very common around very metabolically active neurons, like the interneurons that you're describing, neurons that need a lot of energy, which probably comes to them in the form of sugar. And you also mentioned the blood-brain barrier. The blood-brain barrier is also sort of similar to a perineuronal net in the sense that it's some sort of physical barrier whose tightness and porousness needs to be regulated, because the brain cares very much about what's coming in and out of the brain into the brain from the bloodstream. So if I had to very coarsely summarize what I'm hearing here, it's, the blood-brain barrier has a certain level of integrity that needs to be maintained to it. This high-fat, high-sugar diet can start to compromise that integrity. Within the brain itself, there's also these tiny little things called perineuronal nets, which sort of protect, almost like shells, I guess, like I said, some synapses. Those can start to break down, and that breakdown is somehow due to inflammation in the brain that is caused by this diet.
Yeah. So we set up all these vicious cycles, and it does become very sort of reciprocal in a way. And this doesn't just apply to how neuropathologies are setting up. I was previously doing some work with Dr. Cassandra Lowe at Western University, and we were really thinking about how, when these diets start to change on sort of more of a macro scale, the function of the prefrontal cortex, that our diets change our behavior, and that then we set up these vicious cycles of they can compromise the functionality and integrity of our prefrontal cortex. Our prefrontal cortex is vital for overriding specific behaviors that potentially we would naturally go towards having the sort of resistance of temptation of buying that cake when you get your coffee in the morning, and that it could be that by reducing the functionality of the prefrontal cortex, that there's both the balance of the behavioral control becomes compromised, but then conducting more behaviors that further compromise behavioral control, and this then sets up these behavioral cycles that are very difficult to break. And also some work by Laura Corbett was looking at how high fat and high sugar diets, or high fat diets, I think she was giving rats like condensed milk, sort of like sweet milkshakes, and she found as well that striatal and frontal circuits, which were involved in habit acquisition, these rats start to form habits faster than their companions that are fed a healthy diet. So it's all this, learning and how we behavioral control and these sort of vicious cycles of potentially why not only, we reinforce these behaviors, they become very hard to break. And also for people, who have got caught in these cycles, suddenly large shifts in diet can be so difficult for them and stressful. And we know as well that stress makes us crave these foods as well. So there's so many different things at play beyond, just the simple, microscopic view of potentially our Pavlovin neurons that are starting to, fire or break down and neural circuits. And this spans all the way through to behavior.
Yeah. So these vicious cycles that you're talking about. So literally the very circuits within the prefrontal cortex and elsewhere that give you the power to resist things and to inhibit certain actions are some of the circuits that are specifically vulnerable to atrophy, literal atrophy, if you shift your diet to like this high fat, high sugar diet. So the same circuits, in other words, that allow you to resist a diet themselves break down if you start to engage with that diet on a regular basis. And then of course, you're trapped and you probably feel like you're at the mercy of the food or that you're possessed. And in some sense that literally corresponds to parts of your brain that have physically changed. One of the things I want to ask you about to connect some dots here, we're talking about, it's obvious that we're talking about food and we're talking about diet, but you've already said some interesting things that remind me of something else that might not be, that people might not immediately connect to this, but I'm wondering if there is a connection. So we've been talking about the hippocampus and the prefrontal cortex. We've been talking about diet-induced changes in this case that allow certain parts of those circuits in the prefrontal cortex to atrophy, let's say. So connections are being lost. Not getting lost in the details, we know that outcome, the atrophy of certain connections in the prefrontal cortex, is also very closely associated with other neuropsychiatric states. So one example is depression. Another example is addiction. Depression, one of the hallmarks of depression is a loss of connections, atrophy of certain connections within the prefrontal cortex. And one of the exciting things about some of the research that you're probably also familiar with based on your background is that many of the psychedelics and other so-called psychoplastogens actually do two things, it seems. One, they stimulate very rapidly the growth of new connections. And we know that they do that in places like prefrontal cortex. And we also know that many of them are very potent anti-inflammatory agents. So is it a stretch to wonder if these molecules might have some role to play in the world of diet?
Yeah, it's something I think about on the regular. I'm obviously really excited by psychedelic revolution and being sort of able to be at the forefront at Cybin, looking at novel molecules that are being developed, but also thinking more widely about the utility of psychedelics and their huge potency in terms of their ability to effectively rewire the brain and boost these neural circuits and connections that maybe have become very much hardwired that underpin these kind of maladaptive behaviors. There's these studies looking at smoking cessation. Nicotine is hugely addictive and has such an impact negatively on people's health and well-being, but people are so addicted to smoking that these kind of behavioral responses are incredibly difficult to override. But when you look at the studies that are coming out that are showing 60% of people after one session with psilocybin are able to quit smoking effectively for I think 6 months when they did the follow-ups. these drugs are genuinely causing huge changes in the brain. And it's definitely not difficult to extrapolate from substance use, nicotine addiction, alcohol use disorder, these kind of maladaptive behaviors that are, again, hardwired in terms of associations, associative learning. They're backed up by these very strong associations, difficult to break. That then, how are these how are these drugs potentially able to change other sorts of behaviors, be it binge eating, or people on the other side of the coin, people with anorexia. I saw that there were some phase 2 trials or phase 1 trials, or looking at how psychedelics could potentially start to treat these sort of maladaptive eating behaviors. And again, when you said about the neuroinflammation, these serotonin agonists are incredibly potent, and even potentially at sub-psychedelic doses have prolonged effects that are anti-inflammatory. So thinking about how we have a population that's aging, and dementia and Alzheimer's disease is increasing with longevity and going to place incredible burdens on society, that having these kind of therapies and medicines available that can not only hit boost plasticity but also reduce neuroinflammatory responses that are potentially underlying Alzheimer's. There's these big links between diet and Alzheimer's. I think sometimes people say Alzheimer's is type 3 diabetes. That again, we're seeing changes to the hippocampus by these diets, but it's again not like unthought of, that these states of low level neuroinflammation set up by poor diet could be precipitating or exacerbating the sorts of neuropathologies that are associated with dementia. And I think it's such an exciting field, and I love the term psychoplastogen. David Olson, I think that it's already these drugs are potent. These chemicals can cause long-lasting changes to how our brain is wired, and there is obviously a lot of utility in so many neurodegenerative disorders, but also for anything that's underpinned by a maladaptive behavior.
Mm-hmm. Yeah, I think some of this stuff is really important to emphasize for people, because it's really easy to get caught up in some of the specific studies and some of the specific headlines about psilocybin for depression, or this drug for addiction, or that drug for this. But I think the important point here for me is actually that many of these neuropsychiatric problems, even though they manifest in different ways, many of them at least have a component that's shared in common across them. So, the same kind of atrophy and the same kind of circuit in the prefrontal cortex can subserve or contribute at least partially to one person's depression and another person's addiction. And that atrophy may have been initiated by the same kind of just general brain inflammation that many, 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 many people are experiencing right this very moment. So it's not like these drugs know like what your psychiatric condition is that there could be these common, very general mechanisms that they speak to that have to do with these inhibitory and other circuits that you're talking about in the prefrontal cortex. The other thing I want to ask you about here is, you've mentioned that some of these circuits can be vulnerable because they're close to places like the blood-brain barrier. You've mentioned that some of these neurons that are relevant here that have an important role to play in inhibitory control, generally speaking, they're very metabolically active. And another way of saying that is you gotta invest a lot of juice into these neurons to keep them working properly. And generally that means they're also more vulnerable, right? If they're sort of high octane neurons that require a lot of energy, it's going to be, relatively easy to break those if they're deprived of the kind of energy they need. Another thing that connects those things that I want to ask you about is, we've implicitly been talking about all these things in the adult brain, but some of these circuits are some of the last circuits to mature in the developing brain. And I'm wondering what you can tell us about the role that this diet plays in, say, the adolescent brain or the developing brain, because I'm guessing you can tell us something, and I'm guessing it's not going to be a good thing.
Yeah, I'm like, we know from neuroimaging studies initially that the brain, the prefrontal cortex is the last part of the brain to fully mature. And we also know this from just general behavior of teenagers, that as you embark upon puberty and then going through your teenage years, that you do tend to show behavioral traits that tend to be sort of impulsive. You're not the best at making decisions, and this can manifest not only as reckless behavior, but also making risky choices, but also having quite a high threshold when it comes to reward and reward-seeking behaviors in our modern day environment, replete with lots and lots of high fat and high sugar rewarding foods. It never surprises me when you walk past a fast food restaurant, you've got teenagers sat outside or inside in groups, hanging out at the mall, eating burgers and milkshakes and all that kind of food in big gangs. I'm like, there you go. They're not drinking. But I think that we know that the prefrontal cortex is a late area of the brain to fully form. And we also know that our pulvogular neurons, which are surrounded by perineuronal nets, the last area of the brain for our perineuronal nets to surround these pulvogular neurons is the prefrontal cortex as well. And from my work looking at both adult and adolescent mice that we exposed to these high fat, high sugar diets, we did see changes in terms of the distribution of the perineuronal nets, and other people have as well. So basically really showing that these nets are late forming, and potentially that high fat and high sugar diets may, if they're consumed during our adult and our adolescent years prior, and childhood, that they may start to really derail how our prefrontal cortex becomes hardwired, because through neurodevelopment, that it could basically recalibrate the brain a bit, so that maybe you start to set in turn these maladaptive behaviors where you may have these habits, and then because you have these habits to do with how you eat at a younger age, that they become much stronger, hardwired into our neural circuitry, and that may make then these behaviors harder to break. What else I think is really interesting is for us, we notice as adults, if we eat badly or we start really over consuming a lot of calories, that it shows physically on us nowadays. And I mean, I think I could just sniff some crisps and suddenly my jeans don't fit as well. But for young people, often going through these growth spurts through adolescence, and this requires a lot of energy. And in the same way we saw with mice and rats, when we put them on these diets and we're weighing them from when they're juvenile through adolescence through to adulthood, when they're on these high fat, high sugar diets, it almost masks the actual weight gain in these animals. So because their metabolic rates are so high that you don't see it in the same way. And I thought this was really fascinating because, this sort of feedback typically people are getting. And it could be that, if you're not getting this feedback, kind of socially, or, even in terms of your body image as an adolescent human, you can also be, kind of negligent of your diet and just think, oh, it's okay, it doesn't matter. Like, I can eat whatever I want and it's fine. But your brain is vulnerable at this time. Not only is it, still maturing, particularly in the prefrontal cortex, but it, in terms of your calibration of your reward system, has the potential that your brain expects higher thresholds of rewards. You're always going to overconsume. There's this idea of this, hedonics, and that, potentially you see changes in the brain, particularly around dopamine, that when you overconsume these high fat, high sugar diets, and your neurons start to increase the numbers of dopamine receptors on the neurons, which makes us consume more food to get the same reward out of these foods. And that could be, one of the things that's driving obesity. And when we think about adolescents and teenagers that are consuming these diets, that potentially that is, again, potentially changing the balance of the brain. And there's other researchers that have looked much more deeply into how the dopaminergic system is changed by the consumption of these diets. But Paul Kenny has, he fed rats cheesecake, and they showed behavioral and changes to the dopamine system. But I think that it's important in terms of education for young people, to understand that what they're doing in their younger years may have effects down the line. And I think that health promotion is really important for young people as well, because they're very receptive to the environment, and they're also in a situation where their learning pressures are high. So if young people have, again, when we were talking about the hippocampus being compromised, but only under these situations of increased demands, that what could be more demanding than when you're young and you're like trying to learn everything at high school, or you're trying to learn a new language, you're trying to learn all of your history. And
I mean, I have so many questions that cascade from this, and it's hard not to start to think in quite a dystopian manner about some of these things. But what I was starting to think about as you were speaking just now was, so on the one hand, we've talked about all of these diet-induced changes in the brain that can literally degrade circuits in the prefrontal cortex, or in the case of the developing brain, prevent them from being, built up and worked out and strengthened, so to speak, in the way that they would otherwise. And these circuits are very important for what we'll just call higher order cognitive control, sort of the fanciest, most sophisticated stuff that we associate with the human brain. And so if those diet-mediated deficits are happening, and then on top of that, you've got just the other trappings of modernity that certainly aren't helping our attention spans. I think it's pretty self-evident to most people that, having the social media apparatus that we have on our phone and having a million choices on Netflix, our attention spans are literally degrading. So we've got this sort of surplus of options that are giving us an external decision-making apparatus, right? The algorithm can decide for you. You don't have to decide. You're talking about these diet-induced changes in the brain that prevent some of those circuits that would do that kind of thing from developing or from being maintained. I wonder if at least partially, some of the diet-induced changes to the prefrontal cortex and elsewhere that you've been talking about, is it conceivable that they're contributing to things that we might not otherwise think they contribute to? So what about something like ADHD and attention deficit disorder? Is it possible that diet-induced inflammation might be contributing to that. Is there any interesting correlations or relationships between, say, obesity rates in children and ADHD and things in that realm?
It's not really my area to have looked at, but knowing that ADHD attention is so reliant upon dopaminergic signaling in the brain, and that with ADHD that the typical Ritalin, the methylphenidate, that is important for normalizing the dopaminergic system as a treatment for it, could be one of the things that is potentially changed by the environment. I'm not going to say diet by itself, but it is incredibly interesting that our attention system is so controlled by dopamine as well, and that we now do tend to have a much shorter attention span just because we have so much information incoming at all times. I don't think it's necessarily our fault. Even our brain is just paying attention to so many things. There's low levels of information coming at us from all angles, and I try and think when you have those periods where you're like, just have to do some deep work and concentrate on something, that you do still have all these distractions. Like your phone will start ringing, and then your internet comes up with an email, and there's all these temptations as well to behave in a certain way. And that also gets me thinking, I'm just on a bit of a roll now, that if we have so many temptations for other things that are these almost like micro rewards, like looking at Twitter, or scrolling through Instagram, or looking at TikTok or whatever, that these again are tiny rewards that again we can find difficult to stop ourselves from engaging in, because we do find them rewarding. But potentially because our environment is so awash with rewards that everything is a bit almost blunted, because everything's rewarding. Mm-hmm. And how our brain is potentially just not getting the same kick out of things. And that's why we are scattered sometimes, and we do find it difficult to concentrate. And I find if I want to listen to a podcast, I take the dog for a walk, and I'm just kind of enjoying listening and walking with my dog. But at the same time, I'm like, well, why am I not paying attention to just what's outside? But if I'm home, there's too many distractions. I could be looking on Instagram, or I could be checking my email, or feeling guilty for all the emails that I haven't responded to, or thinking about something else. And it's that sort of divergence of information, and our brains, I think, potentially can't cope with the incoming information that we're required to attend to. It's doing its best, but potentially it is overwhelmed.
So we've discussed how, a certain change in diet, which is eating a diet that's higher in saturated fats and sugars, whether it's in humans or whether it's in laboratory rodents, basically has a negative effect. It has a pro-inflammatory effect in the brain, and that has other consequences, which we discussed. The flip side of that is to simply ask if there's diets that can have the opposite effect. Are there any, and there's lots of stuff out there on the internet about, brain foods and the foods you should be eating. I want to ask you, how much legitimacy there is to some of those claims. But in general, if there's foods that can promote inflammation in the brain, it's perfectly reasonable to suppose that there's foods that at least don't promote inflammation, potentially even foods that have the opposite effect. So what can you tell us about that?
Yeah, so nutritional science is vast, and I also want to sort of preface this with what a rodent eats, it isn't, going to have the same sort of effect necessarily as it would in a human setting, because if you are giving an animal a very controlled diet, which could be supplemented with something, it might be at a much higher concentration than a human could necessarily eat if it's in these kind of supplements. But I think that there's good evidence that a number of compounds which have been around for a very long time are potentially beneficial to the brain. And omega-3s, which are the building blocks of our neuronal membranes, and they maintain neuronal fluidity, which can become compromised when plasticity is decreased. There's good evidence that a diet that has these omega-3s that can be obtained from oily fish are beneficial for the brain. And studies using diets such as the MIND diet, which is based around the Mediterranean diet, good population studies as well have shown that these diets do improve both mood and cognition, particularly in older adults. And there's a lot of work going on out of Deakin University with the Food Mood Center there, led by Felice Jacka. That's they call it coined nutritional psychiatry, and that really using these diets as an intervention for potentially dementia, cognitive decline, and depression. So I think there's that. We also know that there's a number of compounds that occur in nature that are anti-inflammatory, and whether these compounds are being tested particularly for cognition and brain health is almost like supplemental to the fact that these diets and certain supplements can be good for, say, systemic inflammation, such as in arthritis, where your joints are becoming inflamed. So there's also antioxidants, and we all think about blueberries being superfoods, but they are rich in anthocyanins, and these compounds are shown to be anti-inflammatory, at least in certain laboratory conditions. And there's other supplements around curcumin, which is found in turmeric, is also known to be a potent anti-inflammatory. We've got, we all know that a cognitive stimulant that, a lot of us rely on the daily, caffeine, that can, potentiate, plasticity in the brain, stimulant, making us work better. And also, I think that we neglect physical activity, and aerobic exercise is so effective for boosting plasticity, and this works through numerous mechanisms in the brain, but particularly improving BDNF, which brain-derived neurotrophic factor that in the hippocampus helps form memories. But aerobic exercise, although you can't necessarily say, okay, I'm gonna run on the treadmill and burn off 400 calories, this is very a minor amount in terms of your energy expenditure. It's the saying you can't outrun your fork, because you could go have 2 beers and a glass of wine, and that's quite easily nearly like 500 calories of empty energy that you're putting into your body. You can have a chocolate bar and that's 200 calories, and you'd be like, oh, I just ran on this treadmill. But what is important with the utilization of exercise is that if you are looking at those lifestyle changes and embarking on making new habits that are healthy, trying to override some of those prepotent responses when it comes to eating cake or always having dessert, or your general diet that is full of these rewarding high fat, high sugar, unhealthy, hyper processed, ultra processed foods, that by boosting plasticity in the brain through aerobic exercise, potentially you are then going to be able to start to more effectively override these habits. And I think that that's an important component for people who are embarking upon lifestyle changes, is that integrating exercise, it does make you at least feel kind of accomplished after going and working out. You get that sort of feedback of, when you do start improving, that you're like, oh, I started off doing this weight and that feels light now. And it's not because this, 20-pound weight got any lighter. It's just because I got stronger. And, starting to, through exercise and diet, reduce adipose tissue. So then you're putting less strain in terms of neuroinflammation on your brain that's being generated systemically from adipose tissue. And exercise is also increasing blood flow and helping in that respect, and getting these nutrients to your neurons. And there are these taking again into the terms, neuroplasticity being so important for being able to effectively adapt to a new environment. When I think about, again, when we were talking about psychedelics earlier, people who have depression or addiction disorders, they're quite often trapped in these maladaptive behavioral circles, really, that they find it difficult to break. For instance, people with depression, the sort of rumination of thoughts over and over that can make these feelings and behaviors hardwired. And we also know that depression is associated with anhedonia and decreased plasticity. If we start integrating in plasticity augmenting exercise, that then can start to help reverse these stagnation almost, where we're stuck in certain ways, and start to adapt these new behaviors.
One of the things I do want to ask you about, because you mentioned at the beginning that you had previously studied the endocannabinoid system, is cannabinoids. And they're interesting in the context of what we've been discussing for two reasons. One, we know the endocannabinoid system is a very important component of hunger regulation, generally speaking. Many listeners will know first or secondhand that THC is famous for its ability to it actually has a remarkable ability to modulate the palatability of food. I'm always impressed by its ability to do that. So it can make things taste better. It often causes people acutely, although not chronically, to increase the intake of certain types of foods. But also many of the cannabinoid molecules, pretty much all of them, tend to have some kind of anti-inflammatory mechanism that they engage in the body. And so I'm curious what you can tell us about the endocannabinoid system with respect to modulating things like palatability and food intake, and whether there's some connection there with the inflammation side of things.
It's been a while since I've thought about these kind of things. We definitely saw, when I was, I was studying CB1 receptors and using antagonists. So the antagonists were, the idea was that they made food less palatable to people. So rimonabant, its main idea was that it's going to block CB1 receptors in the brain, preventing the endocannabinoid tone. And then this would make palatable foods less palatable to people as a diet type tool. And the pharmaceutical industry has really, struggled with the development of successful anti-obesity drugs because they either have very pronounced side effects, but the newer GLP-1 type system drugs, they are showing a lot more utility. But around the time that I was looking at the block the palatability of the sucrose solution, and understanding that THC, it tends to make people want to consume particularly high energy starchy foods or sweet foods. So these are foods that we already know are quite rewarding, like bread, marshmallows, candy, that kind of food. But obviously this is an important endogenous system in the control of consumption. And also, thinking about how the endocannabinoid antagonists, the major drawback of them wasn't that they reduced the palatability of food. It was that they kind of induced the reduction of palatability of everything. It wasn't specific to food and palatable foods. And I think that the problems that were reported was depression in people, and that potentially suicidality, because people were like, oh my God, like not only does everything taste of cardboard, but the world has become gray as well. So if you start messing with this system, I mean, blocking your endocannabinoids isn't good.
Yeah, that makes sense. It's not like CB1 receptors are only found in the tongue. So if they're everywhere, you kind of have this very general effect. And correct me if I'm wrong, that drug, so if I remember correctly, when I first learned about rimonabant, it's a CB1 receptor antagonist, as you mentioned. It does have the intended effect, as you just said, of reducing the palatability of food, but its consequences, generally speaking, were so sort of broad spectrum and unacceptable that I believe it was taken off the market, and it's really not used at all.
Yeah, so it was, we used it before it was removed from the market, but I remember also another student in lab at Cardiff was having a look at actually the negative consequences of rimonabant, trying to look at whether it started biasing animals towards punishment in really how it is changing affective type behavior, or even making choices for rewards. They start to rather than an animal that would normally make a lever press, you can do a discrimination discounting type assay with rats, where you have your two levers and one has a small reward and one is associated with a large reward. And you can start to, the rats initially are like, well, I'm going to go for the large reward because this, pressing this one lever once gives me 4 pellets. And if I press this other lever once, I get 1 pellet. So I'm going to go for the big one. But you can start to put in different, modifications to the demands of getting the bigger reward. So it could be that you make it so they have to make more responses, and rats will tend to continue responding on the high reward lever until it's not worth them responding on it, or you can start to put in a delay and do a delay discounting task, and by increasing the delay on the high reward, is it optimal in terms of your behavior to keep responding to the one low reward, or to wait longer and only press the large reward? And again, just thinking about that, what's interesting is we try to do this with rats on high fat, high sugar diets and high sugar diets, and rats, just regardless, but were really good at this task, and they would titrate their responses, so that we didn't see any differences between the diet animals. But delay discounting, sort of reward discounting, has been shown to differ in people with obesity versus people without obesity. And there's been a range of neuroimaging studies as well, which again links back to the function of the the prefrontal cortex and reward system, that there could be this, profound changes by obesity and diet to the reward system that do make people, potentially make poorer choices, or, overvalue certain rewards or undervalue certain rewards. So again, these behavioral changes. But back to cannabinoids, it didn't work. But we go on these scientific adventures sometimes. But yeah, I think the cannabinoid system, I think it's incredibly interesting, and the use now of cannabis instead of alcohol for I think is it's showing utility for people who want to relax at the end of the day. Potentially, the CBD utility of that for anxiety disorders. I think that it's all really interesting, and also the anti-inflammatory properties of CBD in particular. Not just topically, but potentially for inflammation within the brain. I think that it's all definitely areas that should be pursued and are being pursued, and I'm greatly interested in.
One of the last things I want to ask you about is the potential connection that you started to mention earlier between diet-induced inflammation and neurodegenerative disorders like Alzheimer's disease. And normally we don't think of these things as being connected, but is there potentially some connection there?
Yes, there's definitely connections between neuroinflammation and the development of neurodegenerative disorders. I was thinking about, particularly our microglia in the brain. So microglia are support cells, and they're basically immune cells in the brain that are there to maintain homeostasis and to remove any pathogens that are entering into potentially damage our neurons and functioning of these the brain. And if we're consuming these kind of high fat, high sugar diets, and our microglia they become primed, even if they don't actually start damaging cells themselves, that they can become primed because they've previously been clearing up cytokines and oxidative stress that's there. It's like they're hyper alert. And then what can happen is potentially there's some neuropathology starts to set in, be it some tauopathy associated with Alzheimer's disease, or starting buildup of amyloid beta plaques in the pathological sense. You could have a traumatic brain injury, a concussion. You might get an infection that starts to also set off changes within the brain. And then the primed microglia sort of overreact to these kind of situations. And when they're in this prime state, they become hyperreactive, and it's like they can start to cause basically mayhem within the brain, that they can actually start stripping off synapses from neurons. In the case of Alzheimer's disease and tauopathies, where the neurons start to die, obviously the microglia are needed to go and clear up this debris. But again, that it's just this sort of that they're primed, and this can then exacerbate the onset of cognitive decline, and potentially other sorts of neuropathologies.
So if your general metabolic condition is such that these microglia are in this primed state. They're not necessarily actively doing damage on a moment-to-moment basis, but when they're called upon, they react with such force that you just get this extra collateral damage that happens.
Yeah, and also there's been some recent studies that have shown that the microglia can start to interact with the perineuronal nets that are surrounding the parvalbumin neurons. And then they start either interacting with the net itself and breaking it down, or then they might also be sort of clearing up any sort of breakdown of the perineuronal nets elsewhere, but then they're still reactive. But then that means that these parvalbumin neurons that were previously surrounded by the perineuronal nets are now unshielded from their microenvironment, and these are highly metabolically active, as you said. They like their environment to be nice and controlled, and that they're very susceptible to oxidative stress. So if you've got free radicals, ROS going around, that then these can start to impact the metabolically active neurons and cause them to dysfunction and potentially break down or become damaged, and the synapses become destabilized, and then you might start to see these changes in cognition and behavioral control because these circuits are starting to break down, and it's either due to the pathology from neuroinflammation, and then that has a sort of knock-on effect to the neuroplasticity, or vice versa, that potentially changes to neuroplasticity means that the brain is less able to adapt to a neuroinflammation challenge, and sort of overcome these events that are happening. And I think that it's so important to think about so many changes that can happen in the brain just as a facet of the environment, and that these can then have in a way really pronounced effects on brain function, behavior, and people's wellness.
Well, we've definitely covered a lot of ground, and there's a lot to think about. In many ways, it's almost an inherently negative subject. Are there any final thoughts or words of optimism that you might want to leave people with about the just general topic of the relationship between the diet and what's actually happening in the brain?
I think that firstly, knowledge is key, and it's really important that we start to educate ourselves more about diet and its effects that aren't just physically on our physique, our bodies, but how it's affecting our brain. I think that because of this interest, researchers have started to look more closely at diet interventions, be it Mediterranean diets, mind diets, and being able to potentially, when somebody starts to experience cognitive difficulties or their memory's not as sharp as it used to be, that they can integrate in these kind of changes to their diet without being reliant upon pharmaceuticals. And I'm also incredibly excited by the premise of psychedelics to be able to boost plasticity and augment neuroinflammation in the brain, and potentially have these profound behavioral changes that are basically what underpins so many maladaptive behaviors that people are struggling with. I think that that's really critical. And unfortunately, I think that sometimes you feel like it's too little too late in certain situations where like, oh man, I've got to lose weight and go to the gym and whatever. But also because we know that the brain becomes more plastic when you're exercising and starting to integrate in these kind of behavioral changes. It's like the first step is always the hardest step. And when people start to understand that, I think that you can start making much more profound changes to your health and well-being. And moving away from the idea that, we're going to have this magic pill that's going to fix everything. Change takes work, and scientists are looking at discovering new ways to augment change and new mechanisms to make it better and quicker. But it's not going to be a magic pill overnight.
Mm-hmm. Well, Amy Reichelt, thank you for your time, and I really appreciated some of the knowledge that you shared with us today.
Thank you so much for inviting me to talk to you.